Okay, everyone, let's get started. We have an exciting day ahead of us with an enormously fantastic lineup of speakers and poster presenters. So good morning, everyone, and welcome to the third Variant to Function Symposium here at the Broad Institute. I'm really excited to see so many familiar faces. How many hugs this morning were already given? It's incredible. It's lovely to see that we have such a strong V2F community, and also lovely to see so many new faces for everyone here in the auditorium in person. We have multiple overflow rooms at the Broad for in-person attendance, and we have over 500 attendees joining us virtually today. So we are 1,000 attendees this year, which is just along the track record of previous Variant to Function symposia, joining from all over the world, spanning academia, industry, venture capitalism, and beyond. And that kind of scale and reach and diversity is really what makes our community powerful, I think. And I'm filled with joy and pride and excitement about that. So this symposium, as the previous symposia of V2F here at the Broad, is hosted by the Novo Nordisk Foundation Center for Genomic Mechanisms of Disease, which is a bridgehead center focused on bridging science and innovation for translation in the metabolic disease space, and really for connecting Danish science with scientists here at the Broad Institute. And as a matter of fact, we had 45 Danish collaborators flying in over the last 2 days here to join us for our Variant to Function Symposium and the annual NNF Center Symposium. And I just want to say the warmest welcome to those I haven't given a hug yet. And I'm so excited that you are here and for the coming days ahead. I will just say Bridgehead Center or bridge building. I'm going to pause and say, as an academic in the United States, where science and so much more is under attack, bridge building, I'm very fond of bridge building. I've always been fond of bridge building, but I'm particularly fond of bridge building these days. And I think bridge building across institutions, across the globe, really, between academia and industry, this between science and the public for increased science communication towards the public, and bridge building across scientific disciplines has become so important. And the bridging of scientific disciplines, I think the V2F field really is a stellar example that has showcased over and over again that the bridging of different expertises, clinicians, biologists, mathematicians, computer scientists, geneticists, patients, and so forth, make the difference and can have an enormous impact. So let's think today as a day of bridge building. Reach out to somebody with expertise outside of your comfort zone. Don't lay back, but engage actively in the discussion. I think Eric Lander and Mark Daly have done a great job last year with their keynote discussion rather than a keynote lecture, in the sense of this is a dialogue. We have to advance the field together as we are moving forward. Also, post on social media so we can engage in discussion with the outside world, even beyond the attendees of this symposium, and actively contribute to science communication towards the public. Great, we have more people trickling in, which is lovely. I have the great honor now to thank the most important contributors for today, and I'm going to start with Kasper Lage, who's here right in the front seat. Kasper has been co-chairing the Variant to Function Symposium with me from the get-go, which has been an enormous pleasure. Kasper is the managing director of the Novo Nordisk Foundation Center of Genomic Medicines here at the Broad. And I think I speak on behalf of the leadership that we are very grateful for your careful shepherding. And I'm also really grateful for your faith and the support in continuing to embark on Variant to Function as one of the guiding principles for our center. So let's give an applause to Kasper. And I will say that the welcome remarks are on behalf of both of us, as we want to thank particularly so our generous host, the Novo Nordisk Foundation, and particularly so Tanja Petersen. Oh, no, there you are, lovely. And Christine Williamson, our contact persons at the foundation. It's so lovely to see you, as usual, an enormous pleasure to interact with you, and I'm very much looking forward to connecting this week and to discussing science and beyond. And this here is actually a picture from your last visit at the Broad in the genomics platform. Arguably most importantly, a huge thank you and shout out to the center's admin and operations team led by Annika Weimar and multiple other attendees, but particularly so Carly Corsano, who has tirelessly worked on organizing and preparing for this event. These symposia, they don't just happen. There's a lot of unseen work in the background. And I would love us to clap and give a huge applause to both Christine, Tanya, and as well as Carly and Annika. We are very grateful. So I'm also going to give a shout out to Todd Golab, the Broad's director, and thank him for his strong support and guidance along the way, and to Eric Lander, who cannot join today because he's celebrating with the family the Jewish New Year. Happy Jewish New Year to all of those who are celebrating, and your families, of course. Eric has been arguably so one of the earliest advocates and supporters for Variant to Function and the Variant to Function initiative here at the Broad Institute, as he has asked me, along with Jesse Engreitz and Aviv Regev initially, to launch a broad internal Variant to Function initiative in 2017, which has been an enormous pleasure. And I'm really excited that Jesse is not only here in person today, but he's also going to share his continuing amazing work in the V2F space later today in this symposium. The V2F story for me is a personal one. Growing up in the Alps, I really have never imagined, I actually grew up on a farm, that I would spend my career connecting genetic variants to disease biology in the context of cardiometabolic disease. But back in 2007, when the first waves of Jivas just hit us as a community, I attended a symposium in Munich, and Thomas Illig was there. And Thomas is was one of the senior authors on the diagram consortium, investigators, who was part of the 2007 largest genome association study back then, or first genome association study for type 2 diabetes published in Nature. And he described the challenge. He said what's expected is that these GWAS signals for diabetes, they come in LD blocks because of the haplotype structure of the human genome. But what probably not everyone has expected is that the largest chunk of these signals is in the non-coding part of the genome. That problem in 2007 hooked me. I took on a PhD project with Thomas and Hans Hauner at the Helmholtz Center and developed a computational and experimental strategy for my PhD thesis in the variant to function context to predict which regulatory variants would be functional and related to type 2 diabetes progression and manifestation. So I'm saying this, it actually took 7 years until this work got published in Cell, which is just an indication of how long variant to function studies, particularly in the past, have taken. And that is being changed drastically now. The point I wanted to make is that at the time, I could hardly imagine that we would be at one point able to trace the full path from genetic maps through mechanisms to medicines. And yet that's exactly what the field has embarked on and is making possible. And I think speakers today, like the Trey Baser and V2F, George Church, Vijay Sankaran with his, as one of our variant to function pioneers, seeing his V2F target moving all the way into the clinic, Ben Kleinstieber and others are definitely examples of the success that genetic information can be moved through the angle of mechanisms all the way down to medicines to treat patients. And with that, other than in the previous 2 years, we launched the V2F Symposium open to the public in 2023, where we focused on genetic discovery and gene regulation and cellular phenotyping. In 2024, we then doubled down on technology development for scaling variant interpretations. That was last year, along with a fantastic keynote shared between Mark Daly and Eric Lander on where human genetics future, or where they're seeing the future of human genetics. This year, our focus is twofold. On the one side, we will continue and double down on scaling variant interpretation along the axis of biology, being gene regulation and cellular programs. And on the other side, for the first time, we're going to reach toward translation and connecting these insights to therapeutic hypotheses. And speakers like Robert Planch, I'm so excited that you are with us here today. Vijay Sankaran and Glenn Kleinstieber are northern lights in this path to literally leveraging genetics to actually translate information into the clinic. Now, since this year's theme is very much about reaching towards translation, it's worth pausing and asking what actually makes a good drug. And I borrowed here an analogy from Matt Nelson, the CEO of Gentians, who has given a fantastic keynote lecture at our MPG seminar at the Broad earlier this year. I'm actually coming to this in a moment, sorry. For now, I'm just going to make the point that at the basic level, what makes a good drug, we can say it's important that it actually works in humans, ideally, not only in mice. For the intended disease indication, it needs to be safe, of course. It has to reach the right place and the right dose, and it needs to be selective, targeting the right mechanism with not too many off-target effects, right? And for the future, the notion is definitely that it has to be mechanistically anchored in human genetics and biology. So we have causal evidence in hand for a given target and differentiate and ideally tailor to patient subgroups. And here's where I'm going to borrow Matt Nelson's analogy that he presented at the MPG retreat earlier this year. He compares it to space exploration, which itself is ambitious, risky, expensive, and has a high failure rate. But he makes the point, a very valid one, that there's an estimated more than $2 billion per commercial launch, which means, as he calculated, that we can fund roughly 25 Falcon Heavy space launches for the cost of launching just a single successful drug. We just have to pause and realize that bringing a given target to the market and treating patients is an enormous expensive effort. Now, what's the problem here? The high failure rate, I just discussed this morning with Church together too, is on the one side the problem of innovation, in that many of these medicines, they fail to measure improvements above the standard of care, since they are often targeting similar pathways. On the other side, and I'm sure Robert will talk about this later this afternoon, it's a problem of attrition, and the attrition problem particularly factors in late stages of clinical trials in phases 2 and 3, which drive the high costs in drug development. That being said, I love this recent work that Matt Nelson and Eric Minical published in Nature last year, where they basically looked at the estimated relative success for a given for a set of targets supported by genetics compared to controls across the different phases of clinical translation and across a whole number of different disease indications. And what we can draw from here, which this community is very well aware of, is that genetics absolutely matter. Genetic support increases the likelihood for a given target to end up in the clinic by 2 to 8-fold. But the particularly important point here is that the most critical impact is actually in the late stages of clinical trials in phase 2 and 3, where exactly this attrition problem of failure at the level of efficacy and safety kicks in. So, thinking about genetics as the foundation is a very meaningful concept. Now, we have talked about this iteratively over the last 2 years. Human genetics has been incredibly successful to identifying hundreds of thousands of genetic regions in the human genome that associate with a whole series of different traits and diseases relevant to humanity. Not only diseases and traits, but also we can start mapping disease progression. We can start parsing individual, large clusters of disease into subtypes and endotypes of disease. It's an incredibly exciting space, and particularly so with increasing cross-biobank analyses, as, for example, the BRAVA consortium is undertaking in the rare variant space. I think Ben Neal is here and will chair a session later. Hi, Ben. Lovely to see you. And the global biobank meta-analysis are spearheading examples of exactly what has to happen right now, which is the meta-analysis of large biobanks across the globe, such as the GBMI undertakes, and meta-analysis 24 biobanks across more than 2 million individuals to come up with robust signals and really drive us towards saturation and lay out the map of genetic associations with disease. We can say that genetic discovery itself from first principles is a solved problem. We have to double down on it, and we have to reach saturation, but we know how to do it very, very well. And we have an enormous amount of genetic signals to work with. The challenge is a different one. That's the reason that we are coming here together every year, where we are looking at these robust maps between genetic variation and disease and traits in humans. And I like to say, you have heard me saying this for those who have been at some of my previous talks, that this is to me the foundation for targets and mechanisms of actions that are actionable towards therapeutic translation. This middle, the mechanisms that enable the translation. But that's exactly this prosecution of genetic signals to disease-driving or disease-modifying biology is what's lagging behind. The biological interpretation is drastically lagging behind. So one could argue that one of the grandest challenges in human health is the scalable mechanistic interpretation of genetic data to lay the foundation for rationally designed medicines. And I thought I'm going to quickly walk you through a few of the challenges that continue to exist in the community and that we have to continue work on and that are being touched on throughout the diverse talks we have in this day, and also an enormously exciting poster session at the end of the day. One of the challenges has always been and still is, I shared with you my story, how I started my PhD, is the fact that the majority of these common genetic signals are located in the non-coding genome, in the form of LD blocks that are fine mapped to some extent, but still in many cases, we are not sure what the causal disease-driving variants are, which target genes they are, and which pathways they implicate. We will have a lot of talks focused on this today. Bridging this gap requires, and that's a challenge in itself, to work with disease-truthful cellular models that respond in disease-relevant ways. So we have to turn ourselves and really critically evaluate, actually, are we actually working with the right cellular model systems? Are we working with a set of stimulations? Or are we assessing in vivo physiology in a way that is recapitulating at least a large fraction of disease pathophysiological processes? We also need, which is rarely existing in the community, matched multimodal data sets across these kind of truthful disease cellular model systems that let us systematically interrogate disease biology as it progresses over the pathophysiological events of a given disease. We also need, sorry, I've tested negative for COVID and flu, but I've been down sick for a couple of, over the weekend, and I have finally recovered, fortunately. So a challenge also is we need scalable high-dimensional readouts that capture both regulatory and phenotypic effects across all of these different phenotypic modalities that are in coordinated ways impacting biology related to disease. And this is an incredibly exciting space to watch. We need, and there's an enormous involvement of these data that are currently being developed to fuel the next generation of statistical models. You hear Wei Zhu today to talk about her SAGE-QTL dynamic modeling framework at single-cell resolution, only possible because of data that are currently being developed in the community. We are looking at every day a new phenomenal sequence-based model hitting the rubber and being available to the community. You have Johannes Lindner today presenting on the Borzoi model. There are many, many more that are currently being trained on a diversity of different data sets, which is an exciting space. I'll also say that Eric and Mark in their keynote discussion last year, they have called for the importance of comprehensively cataloging effects of effector gene modulation through a large set of perturbative experiments across a wide range of primary disease models. And Elisa Donat, a phenomenal group leader here at the Broad in the Lander Lab, has taken on that challenge to wrangle the groups. We are lucky enough to be part of that effort to get together the community in the disease space and really think about how can we systematically perturb thousands of targets in a harmonized manner with readouts that not only stop at the RNA level, but also fold in multiplex site-seq readouts and optical information through high-content imaging that many of you know we are really fond of, to develop data sets that enable us to develop the next generation of computational models that would be able to identify, sets of GWAS hits that converge on the same functional pathways, for example. And finally, I think a space that is incredibly exciting to think about is ways that are often called, or the strategy is often called Jivas in a dish, where we are looking at systematically mapping genetic variation to disease-driving biology in these truthful patient models. The idea was incubated as part of the International Common Disease Alliance, together with Gosha Trinka at the Sanger and many others in the community, and called the CellMap framework, where the idea here, I'm going to just include 2 slides of results on our end in my lab, where we are, have these truthful primary patient samples, we are pooling them together and then reading out across multiple different phenotypic modalities to capture the full propagation of biological information in the cell as the cell is exposed to disease-relevant stimulatory conditions. The reason I wanted to include this slide is to show you 2 nuggets of what we can start drawing from it, which I think is exciting. In just a sample size of 118 donors of subcutaneous primary adipocytes exposed to a series of different stimulatory conditions, and reading out with 10x multiome the single nuclei chromatin accessibility, as well as RNA, and using our metabolic disease-oriented high-dimensional image-based profiling lipocyte profiler, we can start seeing loci through a stringent co-localization framework like this one here that robustly associates with fat distribution. And you can start tracking the full path of how genetic variation acts on chromatin accessibility, connected to a co-localized expression QTL and to an image QTL to make the path and the connection of genetic variation impact not only on gene regulation, but also downstream phenotypes that relate to disease biology. When we look genome-wide in these 118 donors across the different disease-relevant stimulatory conditions in adipocytes, we can then ask for the co-localization rate across metabolic disease traits. I have a set of examples here like fat distribution, insulin-sensitive, insulin resistance, and glucose homeostasis and lipid homeostasis. And you can acknowledge that roughly 10% for LDL, where subcutaneous adipocytes are not expected to play a major role, but up to 80% of these Jiva signal appear to co-localize with any of these molecular QTLs coming from attack RNA or image-based profiling. Huge shout out here to Hisham Dashti and Yi Wang in my lab, fantastic scientists, and Yi is a postdoc who have put together these very recent data sets on a short notice. I also want to shout out to Sophia Metz, a great postdoc in the center, co-advised by Camilla Scheele and myself, and previously with Thomas Kilpinen. If you are interested in reading a really good variant to function review in the adipocyte space, please look at this one. It's a great one. Okay, so what's new and coming in variant to function research? Just quick notes. On the one side, context. We learned that context really matters enormously. Disease stimulation context particularly, whether it's nutrient signaling, inflammatory cues, or hypoxia, the cellular environment matters to unfold and manifest variant effects. That's what the community over and over finds these days, and it's important to factor in. We need to read out biology across multiple phenotypic scales. We are also, and that's the second point here, we are entering a biobank scale era of molecular phenotypes, where the 10K consortium, for example, but also the UK Biobank and FinGen are now ramping up to basically map 10x multiome, for example, along with metabolomics and proteomics in tens of thousands of individuals that will be available to the community and make a huge difference in variant interpretation. I also think that this point here, from gene-centric to program-centric view, which my lab is embarking on a lot currently, is an important one, where we are moving beyond linear Jivas variant to gene kind of translation, and going more and more along the notion that a lot of these loci, they act through coordinated polygenic cellular programs, and mapping these cellular programs paves the way to identify patient subgroups towards therapies. And then there's, of course, AI. That's a bucket that is exploding at the moment. I just saw from Zhongsu earlier yesterday their new GPN model, which seems to be, at least from the first glance, phenomenal in terms of enrichment of SNP heritability. There's one model after the other being developed and trained by increasingly better data, in the sense that they capture disease biology better and better, which is an exciting space. I'm very fond of the iterative lab in the loop model, where we are working with challenges in machine learning and thinking about how do we have to design an experiment to then develop a model and go back to improve and refine the model itself in order by designing an experiment that is ideally suited for addressing exactly that problem. And then finally, the translation piece, where of course the patient-specific in vivo gene editing through base editors earlier this year is one of the examples that is just so incredibly exciting to witness in our field. And we have Ben Kleinstieber, who has been involved in this study and is working in this space with such an enormous track record today, speaking to us. Okay, so the sessions, very quickly going through them, addressing multiple of these challenges that I laid out. We are going to start with scaling variant interpretation shared by Dieter de Montes and have exciting talks from Ruth Loos, Jason Flanick, and Wei Zhu, focus on the question, how do we move from millions of variants to interpretable maps and disease endotypes at scale? We are then moving on to the gene regulation session shared by Brad Bernstein, along with an exciting talk from Jesper Madsen and Jesse Engreitz in our center, focused on decoding the regulatory architecture that connects variants to affected genes. And then I think after lunch, we are decoding variant effects through cellular models shared by Tune Pears, with exciting talks from Chloe Villani, who is co-leading the Human Cell Atlas, and Johannes Lindner, who is or has been in David Kelly's lab and developed Borzoi, a really intriguing machine learning model. And finally, the last session is translating variants into therapeutic insights, shared by our very own Ben Neal and the speakers. And I'm so excited, Vijay, that you could make it this year. I don't know where you're sitting right now, but I'm really looking forward to hearing you as one of the V2F pioneers and seeing through all the way to clinical implementation of your work, as well as Ben Kleinstieber, who I've praised earlier. Kasper, I know, will do a phenomenal job at introducing Robert, so I'm leaving that to Kasper. I'm very much looking forward to the talk. So this is kind of the full arc that we are spanning today. I'm not going to repeat all of the different parts, but we'll just say the whole is greater than the sum of its parts. I'm just including here a few icons. I was sick when I made this, so there's a lot of gaps in this slide. I apologize for it. The point I wanted to make is this needs bridge building. The V2F field continues to need bridge building. This is why we are so strong. Along with our V2F Symposium hosted by the NNF Center, I also want to shout out in the spirit of bridge building that the Wellcome Connecting Science is launching the Biology of Scale conference series next year at the Wellcome Campus. That's a symposia that Gosha Trinka, Craig Glastonbury, Jonathan Pritchard, and myself have put together, focused on developing and thinking how we can measure and model biology at scale through new tools and technologies combined with computational strategies.